Good afternoon again. And my title is Southern, Northern, and Western blotting. Since the principle is almost same, I will, after introducing and talking about the principles of techniques, I will mainly talk about the Western blotting. Southern blotting is a technique which allows the detection of a specific DNA sequence in a large, complex sample of DNA, which is named after the British biologist Edwin Southern in 1975. Southern blotting combines two methods. One is the agarose gel electrophoresis for size separation of DNA and transfer the size separated DNA to a filter membrane for probe hybridization. After recognition of Southern blotting for the detection of DNA, same technique is used for RNA, and which is called as Northern blot, and then same technique is used for protein and is called Western blot. And now I would like to talk about the Western blotting in detail. Western blot analysis can detect one protein in a mixture of any number of proteins. It was introduced by Tobin in 1979, and it is also called immune blotting since Western blotting uses antibodies to identify specific protein targets bound to a membrane. There are several steps, and the first step is the separation of proteins using a polyacrylamide gel electrophoresis, and the other step is immobilized protein onto a permanent substrate is called a membrane, and we use nitrocellulose or polyvinyl difluoride membrane for this purpose. And the other step is identified protein of interest. This is our protocol for Western blotting. A high-quality antibody directed against a desired protein is used as a probe to detect the protein of interest. This slide shows Western blotting. As you know, we already have, and we already talk about the protein, how can we get the whole cell extracts protein extraction. After getting a protein mixture, you will load your sample to agarose gel, polyacrylamide gel, and in electric field, it will be migrate by size. Proteins separated on basis of size and electrical charge in electrical field. Then there is several proteins on the gel. After blotting, separated proteins transferred from the gel to membrane, and then you can use the specific antibody which recognizes your protein of interest, and then you can recognize or visualize the protein. The first step of Western blotting procedure is to separate the proteins in sample by using the denaturing gel electrophoresis. It's called SDS page electrophoresis, and the previous speaker talked about that. And those are the apparatus of electrophoresis. Mainly in an electrical field, proteins moves from the negative part to a positive electrode, and the larger fragments take place in the top of the gel, and the smaller fragments moves rapidly move compared to larger fragments and take place at the bottom. Second step is transfer to membrane. It's called blotting. The actual blotting process may be active or passive. Electroblotter is used for faster and more efficient transfer of protein from gel to membrane. The proteins are transferred to a sheet of a special solid matrix. Is called membrane, and there are two commonly used, commercially commonly used membranes. Is called nitrocellulose and polyvinyl difluoride membrane. Sandwich of filter paper, gel, membrane, and more filter paper is prepared in a cassette, which is placed between electrodes, and electric current is passed through the gel, causing the proteins to electrophorese out of the gel and onto the membrane. Nitrocellulose membrane are the popular matrix which is used in protein blotting. They have highly affinity to bind the proteins. Most proteins can be successfully blotted using a 0.45 micron pore size membrane, and for proteins of low molecular weight or peptides, 0.2 micron pore size membrane is recommended. Another solid matrix is polyvinyl difluoride membrane. These membranes are highly hydrophobic, and it's really hard to get wet in aqueous solutions. Before use, you have to use pre-wet in methanol. Due to its polyvinyl difluoride membranes are used for immobilization of proteins due to its non-specific affinity for amino acids. It has 0.22 or 0.45 micron pore size. It's available sheets or roll. There are forms of membranes. And during blotting, we have to use a filter paper. Filter papers are pre-cut cotton sheets for wet or semi-dry, passive or electrophoretic transfer of proteins from STS page to membranes. Sheets of blotting buffer are necessary components of transfer sandwiches and cassettes that typically must be assembled for various kinds of protein or nucleic acids, gel to membrane transfer protocols. There are some buffers for blotting, and common buffers used for Western blotting are Tobin buffer. You can see the formula, and CAPS buffer. I prefer actually Tobin buffer for the Western blotting. Those are the blotting equipments. And there is a lid, electrode assembly, and gel holder cassettes, and cooling unit. You have to use this sandwich model. It's important to get filter paper, membrane, gel, filter member, and foam pad in the cassette, and then put into apparatus and use the blotting buffer and power supply. After blotting, the proteins retain the same pattern of separation they had on the gel, like this. Of course, you cannot recognize your proteins, but you can see the protein marker as a guide. Okay, now I would like to talk about transfer methods. There are four major ways to transfer macromolecules from gels to membrane: diffusion blotting, capillary blotting, wet blotting, and semi-dry blotting. I prefer the wet tank blotting, actually. As I told before, there is a capacity of binding of membranes to proteins. After blotting, the proteins move to membrane, onto membrane. Cellular proteins will bind to the membrane since the membranes have affinity to bind the protein, so antibodies are also proteins, and they can also bind non-specifically to the membranes, not just your proteins of interest. So to control these undesirable binding, membranes have to be blocked using a general protein. We always use albumin for this purpose, which binds to any remaining sticky places on the membrane before using your primary antibody. As you can guess, there are some washing steps I couldn't say before. After blockage of non-specific binding sites on the membrane, it's primary antibody, and which binds your protein of interest. And then the secondary antibody will be directed to our IgG of the species in which the primary antibody was raised. That's why we are calling Western blotting is also immunoblotting. And in almost all cases, the secondary antibody will be conjugated to horseradish peroxidase. And the step 5, horseradish peroxidase is an enzyme that catalyzes the conversion of an enhanced chemiluminescent substrate into byproducts and light. ECL substrates allow for the detection of extremely minute quantities of protein. It works like this. There is a antigen and protein, antigen protein of interest and primary antibody, and your secondary antibody conjugated with horseradish peroxidase, and you can use the ECL, and which after catalysis of ECL by horseradish peroxidases, you can detect by film the light. And I will show some pictures from Western blotting. This is a electrophoresis part. You have to prepare the gel. We use the polyacrylamide gel for the separation of proteins. Washing step, and put into the tank. And STS-page buffer, running buffer actually. And loading to your samples. And during the electrophoresis, you can see where it is. After electrophoresis, remove gel from the gel cast and place into the transfer buffer. And we have to cut filter paper, preferable larger than your gel, and cut a piece of membrane, soak in methanol. As I told you before, polyvinyl difluoride membrane is very highly hydrophobic, and you cannot get wet in aqueous solutions. That's why we have to use, before blotting, a second for methanol, and rinse it transfer buffer. We don't want to use methanol actually after getting wet. Place 3 pieces transfer buffer soaked filter paper on the blotting apparatus and place the membrane onto the paper. During that time, we need no air bubble actually. And place the gel on top of the membrane in the correct orientation. The marker should be on the same side that it was when you loaded the gel. Gently roll out any air bubbles and place 3 more pieces of filter paper on top of the gel. Set the transfer apparatus to 80 volt for 2 and 3 hours, or 8 volt for overnight blotting. This is our sandwich model again. And there is another machine uses a dry blotting actually. It's called iBlot, and uses dry blotting. And really fast, it takes 7 minutes for blotting time. incredible. After blotting, incubate the membrane with primary antibody and wash the membranes with TPBS buffer. It contains twin-20, and incubate the membrane with secondary antibody and wash the membranes again. And incubate the membranes with ECL solution for 1 minute. Wrap the membrane and expose to X-ray films. You can keep your membrane and you can use after stripping with another antibodies. You can continue to another Western blotting. And this is a picture from one of my experiments actually. After Western blotting, you can get the proteins bound. Not only ECL, but also you can use the chromogenic substrate, and you can see the precipitation of chromogenic substrate on the membrane. This is a picture from our previous courses, actually. During the process, be careful. Do not touch the membrane without gloves because, as I told you before, highly affinity to binding proteins. Do not let the membrane dry out and rule out any air bubbles during transfer step. There is no air bubble between the membrane and gel, and be careful to electrode position. The previous speaker mentioned that. It's really important point. And this is my last slide, and thank you very much.